We're now ready for our next speaker. And, you know, one of the things that I love about this conference is we have all these brilliant, changing the world scientists, and they come here and they're very friendly and casual. So it's a great combination. And speaking of friendly and casual, I would like to bring Dr. Sarah Weckhuyzen up to the stage now. Yes, big round of applause for Sarah. So Sarah, I'm going to keep bios really short, but I think the reality is that Sarah is the lead author, and Ingrid is also on the paper of the very first paper that identified KCNQ2 for our families. So I think that's probably all I need to say about her significance in what affects us most. So Sarah, I'm going to turn it over to you and let you get started.
Yes. Thank you, Sarah, for your very nice introduction. And first of all, I want to thank Sarah, Scotty, Caroline, whoever to invite me again to this meeting. It has been a while since I've been here. COVID and babies that interfere with planning. And it's always great to be here and to see the community growing. And as a clinician researcher myself, I can only confirm that it is this kind of meetings and this kind of strong communities that keep researchers focused on a specific disorder, focus on working towards a cure, and motivate them to collaborate. So really, I think this is a great work that you do. And again, happy to be here and talk to you a little bit or guide you a little bit through the story of KCNQ2. I think many of the parents here have been at previous meetings, and for some of them, this might be old news, but I hope that for some people, this talk can be instructive a little bit. So first of all, I'd like to say that, or say something about the incidence of KCNQ2 related epilepsies. The disorder is rare, but among the genetic epilepsies, it's not that rare. There's some studies, for example, there has been a big cohort study in Scotland a few years ago, and it showed that KCNQ2 related epilepsies, and then I mean the whole spectrum, is present in 1 in 17,000 birds. So this is not nothing. Another study made an estimation of the incidence of the severe form of KCNQ2 related epilepsies, and they estimated it to be between 1 to 3 per 100,000 people. Another, I think, very important thing to realize is that about 1 in 3 children with a genetic form of neonatal, so this means neonatal epilepsy, so epilepsy with an onset in the first month of life, has a KCNQ2 mutation. So this is really, it's actually a really important gene in genetic epilepsies. And this is also illustrated, this is very small. And I think it's very difficult to read. This is from a study from a few years ago that went into literature and looked at all studies on genetic epilepsies and which genes are involved. And the first gene, you probably cannot read it, but it's SCN1A. This is the cause of Dravet syndrome, for example. Some of you might have heard of it. Also a very strong patient community. And then the second one here is KCNQ2. And so it's the second most important gene in genetic epilepsies. Very shortly, the history of KCNQ2 related epilepsy, because Sarah very nicely said that we described as first the gene, but it's a little bit more complicated. The story actually begins in 1994, the first gene for epilepsy was discovered, and already in 1998, KCNQ2 was identified as a gene that's responsible for familial neonatal epilepsy. They called it at that time benign familial neonatal epilepsy. Nowadays, they speak from self-limiting neonatal epilepsy. And these were families with children that had seizures at birth and then developed normally. And it took until 2012 when we described that mutations in the exact same gene, so in KCNQ2, are also identified in children with not only neonatal epilepsy, but with neurodevelopmental problems. A few years later, I think in 2017, we and others, like John here, described that some children with KCNQ2 mutations have cognitive problems, neurodevelopmental problems, but no neonatal epilepsy. And then you see 2019 there, that's because we described the exact same thing for KCNQ2. So you see that the gene has been around quite a long time, but the description of the severe disorder is more recent. Now, over the years, our studies have been performed, and one of the things is that people try to study the effect of these mutations. And you can imagine it's very difficult, and I'll come back to that later. So KCNQ2 encodes for a channel, a potassium channel in the brain. You cannot go and just take some brain cells of a kid to study what the effect of the mutation is. But what people do is they make a construct of the gene and they express it in another type of cell. Often it's oocytes from frogs, for example. And then they study the effect of the mutation and the function of the channel in this cell type. And over the years, we've seen that many of these different mutations have different mechanisms. They have a different effect on the protein function. And then the question is obviously, do all these different types of epilepsy related to KCNQ2, do they need different therapeutic approaches? And in this talk, I'll guide you a little bit about what we've learned until now. But first of all, I think it might be good to go back first to the basics. And I'm sure that many of you already know everything about it, but just start with a very basic. So what is DNA? So DNA, it's a molecule that's in each of our cells. And it's like a genetic code. So it's really the code for our cells and our body, how to function. It's like a manual. It consists of a code of 4 different letters or nucleotides, A, T, C, and G. And the DNA of each of our cells, DNA is packed in chromosomes. It consists of 3 billion of these nucleotides. So you really get like a book consisting of words of 4 letters. And now what is a gene? A gene is a part of our DNA that carries code for one single protein. Okay, if you keep, I don't know the pointer words. Yeah. So this is the DNA. These are these letters, and 3 of these letters are a code for a specific amino acids, and together these form a protein. Now we have DNA that comes from the mother and the father, and in general, for most of our genes, we have 2 copies, maternal one and a paternal one. And now there's disorders where if you have 1 copy left of your gene, or only 1 copy that works well, then you have a problem. And there's other disorders where you're perfectly fine if you only have 1 good copy of the gene, and you only have a problem when the 2 of the copies don't function well. And then you can ask yourself, what is a mutation, or why do these mutations occur? And I think that I want you to bring you back a little bit to how the human body is formed, and then it's actually not that surprising, because you have the egg cell, you have the sperm cell, they come together, and then this DNA that is in this single fertilized egg, it has to be copied to every single cell in our body. So you can imagine it's actually very surprising that we don't have more mutations. There's a very good control mechanism in our body, but sometimes it fails. And that's how a mutation occurs. And when it occurs during this generation of the baby, let's say the embryo formation, then you have a de novo mutation. So the parents don't carry the mutation. But obviously, when this boy, in this case, he grows up, he has this 1 mutation and 1 copy of the gene, and he can pass that on to his kids. And then a mutation becomes inherited. There's some more complexities to it, mosaicism and stuff, but I understood there will be a genetic counselor today or tomorrow talking about these issues. So I leave it to this very basic. That brings us to KCNQ2. So KCNQ2, the gene, it carries the code. Remember, DNA, it's all about coding. The code for a potassium channel subunit. And sometimes it's also the protein itself called KV7.2. So here you have the DNA and the gene of KCNQ2. It contains 3,253 nucleotides, so these letters. And they carry the code for a protein that contains 872 amino acids. And what does it actually do? Actually, it has a code of a subunit of a potassium channel. And this potassium channel, it's present in every single neuron in our brain. And it's very important in the communication between neurons. This is a figure of a neuron. As you can see, different neurons connected to each other, and they connect to each other through an electrical signal, and that goes all the way down this long part of the neuron. We call it the axon. There are some chemical substances that are released, make connection with the next neuron, and also here an electrical signal is generated. And these potassium channels, they're very important in this process of generating these electrical signals or action potentials. As a matter of fact, the generation is generated by opening of sodium channels, and the potassium channels, they make them like stop. It's like a brake. So they prevent the neuron to be hyperexcitable. And they're very important. They're in every single neuron in our brain. So this is really the big structure. So every little circle is an amino acid. This is the membrane of the cell, of the neuron, in this case, the nerve cell. It goes through the membrane a few times, and then they have a long part that's inside the cell. This is a more simplified way of making a figure of it. And so this is one single subunit, and the channel itself, it consists of 4 of such subunits. Together, they wrap around each other, and they ensure that there is a pore in the middle where the ions of potassium can flow through. And then there's these side chains, and the channel consists of 4 subunits, and they can form subunits only consisting of KCNQ2, but they can also make a channel together with KCNQ3 or KCNQ5. And sometimes the channel can be closed, and then the potassium channels cannot flow through, or it can open. Very important to realize, or what people have studied, is that KCNQ2, it's already expressed in the brain from very early in fetal life. So this only illustrates how important it is for the functioning of nerves. Then again, the very beginning of KCNQ2, it goes back, or when it was identified, was 1998. At that time, it was identified in these kind of big families. So there were families where kids had neonatal seizures, beginning in the first few days of life. Often they were tonic, that means that babies stiffen up, sometimes with some colonic movements, so some jerking in between. Children can turn blue because they present apnea. The heart can slow a little bit, but the seizures remit in the first few days or months of life. And in between, the EEG of the kids is totally normal, and they also develop normal. Most of the time, these mutations are familial, so you see them in big families. But as I just explained, the mutation has to occur at one point. So sometimes you can find these mutations also de novo. Many of these mutations have been studied in the meantime in a cell, so in a dish. And what they've seen is that the mutations that lead to these self-limiting familial neonatal epilepsy, it leads to a loss of function. So the channel doesn't work well. And the current through this channel is about 25 to 50% less than it should be. So one of the 2 copies doesn't work well. And that brings me to 2012, when we indeed described, with others also Ingrid there, that some mutations that do not lead to this very benign or self-limiting neonatal epilepsy, but you can also find them in children with a much more severe disorder. So these children also had neonatal epilepsy, but they did not develop normally. And so this is a little bit the core phenotype or clinical presentation of these kids. So they have neonatal onset seizures, just as in the self-limiting form. Also here, often there's a stiffening with some clonic movements. Also here, often turning blue, apnea, desaturation, bradycardia. But kids have a lot of seizures with a dramatic onset, multiple seizures daily. They're often much more difficult to control. And the EEG in between the seizures, it's not normal. Often you see this kind of pattern. So you have a burst of electric activity, and you have some kind of silencing in the EEG. And this already shows that something is not that well. And there's a range of cognitive outcome, often a severe to profound intellectual disability, although it's a whole spectrum. And so there's definitely kids with milder problems and a range of comorbidities, as all of you here in the room probably know, sleep problems, as Ingrid just also mentioned, gastrointestinal problems, behavioral problems, etc. So again, this is a totally different kind of disorder than the one that was described in 98. And what happened then is that researchers start to look and compare these different types of mutations. And what they saw was that these mutations in these big families, they were spread all over the protein, while the mutations that led to this more severe form, they were really localized, or they were clustered in very important and very specific domains of the protein. In this first loop that goes through our transmembrane portion, where the voltage sensor is very important, here in the pore of the channel, and then in the end, in specific domains that connect with other proteins. So really in very crucial domains of the protein. And think, as many of you might know, this registry that at Cooper, that I think will be here later in the conference, set up, has been instrumental in studying the different effects between these two mutation types. But because this allowed us to, researchers at Cooper and others working on electrophysiology of these channels, to pick specific variants and also test them in this dish, and to compare what the difference is with these benign mutations. And without going into too much details about these figures, what you see here is this is the current through a normal channel, and this is the current that goes through a mutated channel. And you see there's a more than 50% reduction of the current. Now, what does this mean? That means that it's not just that this one copy of the gene doesn't function well, it also interferes with the functioning of the good copy of the gene. And this is something that we call dominant negative effect of mutations. So it shows actually that both types of neonatal seizures caused by KCNQ2 mutations are caused by a type of loss of function mutations, but the ones that lead to the more severe syndrome, they're more severe, they have a bigger effect. There's a bigger loss of function, there's less current going through the channel. Now we've come quite, a long way since then. We've learned a lot. For example, this is a nice study from Tristan that will be here, that first showed in these big families with neonatal seizures that carbamazepine, or sodium channel blockers, a type of anti-seizure medication, they seem to have a good effect on seizures, these neonatal seizures. And how did they show this? They looked at when this carbamazepine or another drug was started and how long these children stayed in the hospital. And if they gave carbamazepine very early, then the children could go home much quicker. While if they didn't give it, or they give it much later, children stayed in the hospital longer, so they had longer seizures. And since then, a similar beneficial effect on seizures of carbamazepine and other sodium channel blockers have been shown also in this more severe form of KCNQ2 related epilepsies. And why is that? Again, why is sodium channel blocker? Remember what I said, these sodium channels and potassium channels, they work together very, very closely. The sodium channel blockers open and start this action potential or the electrical signal, and the KCNQ channels have to block them, and they block the current. And if it doesn't work, there's too much opening of these sodium channels, and you block that with these sodium channel blockers. So it's a quite intuitively thing. Obviously, we cannot talk about treatments without mentioning ezogabine or retigabine, that many of you might know. Ezogabine or retigabine has been a drug that was on the market for epilepsy, and it's a potassium channel opener or activator. And several years ago, colleague Maurizio Tagliatella, he showed already that, again, in a dish, this potassium channel opener, it can revert the effect of the mutation on the channel function. When you give it, the channels open better, and you have more current. And there were some retrospective studies, again by John here, that showed when you give retigabine to kids with KCNQ2 encephalopathy, this was when retigabine was still on the market, it also seemed to have a good effect on seizures. And as you all know, there was an EPIC study that tried to trial this in kids with KCNQ2 encephalopathy, but unfortunately the trial was stopped. But I think we will talk more about KV7 openers later today. In the meantime, there has been a lot of progress by other researchers. For example, we have a mouse model for KCNQ2 developmental and epileptic encephalopathy, one from a French group, and I think again tomorrow there will be other people speaking about other mouse models. And why is it important? These mouse models allow us to study more easily the function of KCNQ2 in the brain, but also, for example, to trial new drugs. But so it seems all clear. We still have a long way to go, but it's really a nice story. KV7 or the KCNQ2 is the break of the neuron. If it doesn't work well, the break doesn't work well, and you have hyperexcitability of neurons, and you get epilepsy. But then it started again by Maurizio Tagliatella, so a collaborator from Naples, who has been studying KV7 or KCNQ channels for his whole career. Is really testing the channel or measuring the currents in a dish. And what he showed is he tested a few very specific variants in the gene that were located at these positions where these positively charged amino acids are located. And what he has seen is that these mutations, they don't lead to a loss of function, but they lead to a gain of function of the channel. So there's too much current, potassium current, through the channel. And this was remarkable at that time. And again, here the Ricky database was instrumental, because when he described this, we went to the database and looked whether there were children with these mutations. And then the first one we studied was this R201C and R201H. So what this means is the R and the C are different types of amino acids, and the 201, that's the position in this chain of amino acids. We collected the treating physicians and parents of these children, and we looked at the phenotype or the clinical presentation. And these children, they didn't have these classical neonatal seizures with stiffening. They had like, call them myoclonus, so jerking, very arrhythmic jerking of the body, often stimulus sensitive when you make a noise or touch them. Later, they developed epileptic spasms, and they had a profound delay. So they had really very severe developmental problems. And then John, again here in the room, he looked at children with another mutation that Maurizio studied. And again, here, apparently the kids with these mutations, they don't have neonatal seizures. They might develop epileptic spasms a bit later in life, and at a few months of age, and have neurodevelopmental problems, but no neonatal seizures. And then last year, we looked at another mutation that Maurizio studied here at the bottom, the R144Q and W. And we saw that these kids, some of them don't even have epilepsy. They have neurodevelopmental problems, often with autism. Some of them develop epilepsy later in life, but definitely not all of them. And they particularly have a very disturbed EEG when they sleep. In general, we saw that the patients with the R144Q were more severely affected than the ones with R144W. And this correlated very well with the function of the effect of the mutation. I mean, you have a bigger gain of function, you have a more severe clinical phenotype. So have we learned something for these kind of patients, KCNQ2 encephalopathy due to gain of function mutations? This has been described more recently, and it's more rare. So unfortunately, until now, we have less insights in how you should treat these children. I think all of you that are on Facebook might have seen parents talking about other types of drugs that are used in other types of disorders, like amitriptyline that's given for headache, depression, donepezil that's given for Alzheimer's disease, guanfacine that's given for ADHD. All these drugs seem to also have an effect on KV7 or KCNQ channels. If they block the channel to some extent, maybe some parents here are trying it. Until now, it's not very clear yet what effect this has on the problems that you see in these kids. But hopefully in the future, there will be more efforts to study the treatment in these children. So this is a little bit a summary. If this is the current through the channel can be normal. You can have a mild loss of function, so less current going through the channel. When it's mild, you just get neonatal epilepsy. When it's a very severe loss, you get neonatal epilepsy with neurodevelopmental problems. And to the other side, if you have too much current, you get neurodevelopmental problems, but normally not with neonatal epilepsy. You get epilepsy later on, or you might not get epilepsy at all. So what does this mean? So it means, again, because we also described there might be different treatments that are effective for the different types of mutations. So when you are in doubt about what the mutation in your child does, talk with your doctor and have him or her or geneticist check whether this variant has been reported already and whether we know what the effect is. You can look in the wiki database that I just mentioned, or if it's not a known, just reach out to people in the KCNQ2 field and ask their opinion. I think as a few final slides, obviously there's many remaining questions or many questions that remain, and that's where research comes in, and Scotty nicely introduced that. For example, there might be kids here in the room with the exact same mutation, but still they're not exactly the same, right? They might have different problems. And we hypothesize that other genetic variants in our genome, in our DNA, might play a role, and that's where this genetic modifier study comes from, and with the swabs. We also don't fully understand yet how these mutations, they lead to neurodevelopmental problems. It's quite clear how they lead to epilepsy, at least for the loss of function ones, but not yet why it leads to problems with cognition, behavior, and all the other comorbidities. We also still don't fully know what the exact or the best treatment is for these children and how much time we have to treat the condition. These are all questions that we try all together to address by working in the dish, in neurons, in mouse models, and obviously also working together with the patient community. I think we're all, or we should be a big team working all together, patients, doctors, researchers, industry, and together we will reach our goal to find a better cure. So this is my concluding slides. So I've shown you that both this dominant negative and gain of function variants that lead to different types of KCNQ2 developmental and epileptic encephalopathy, that it's very important to know what the mutation of your kid does, but that clinical features can be highly predictive. And if you have neonatal seizures, it's very likely that it's a loss of function mutation. And many KCNQ2 variants are recurrent, so there might be kids here in the room that have the same variants, and it might help to talk to each other or to look in the registry. And I said that is my last slide. It's actually not my last slide because I want to remind you that we worked together also with the KCNQ2 Cure last year to write a book, and there's also an online version where not only researchers working on KCNQ2 and clinicians working on KCNQ2 contributed to, but also parents. You can buy the book, but there's also a free online version, and here's a QR code that you can scan. So I think it's a very useful tool for people that want to know more about it, to hear stories of other parents or to read about what KCNQ2 related epilepsies really are. And with this, I really want to thank you. And I don't know if it's time for questions now or at the end. Yes.
You're waiting for something we're going out to you. No, you go ahead and ask. We'll just get in my queue so we can hear so everyone can hear your question. And the other thing I want to let you know is we're getting the mic to Clara. This is there'll be 3 panelists together do Q&A. So if you don't get your question now, you will get it. Go ahead.
How frequent are you seeing somatic anxieties in parents who have no idea and now are going to shock?
That's a very good question. The problem is we don't specifically look for it. So it's very difficult to say how often it is. It has been done in bigger study, because it is not something that's only specific for KCNQ2. It's something that applies to all de novo variants. And Ingrid, for example, has done a nice study that looked at a group of people with de novo mutations in epilepsy genes in general, so not only KCNQ2. And correct me if I'm wrong, Ingrid, but I think you found mosaicism even in 10%, right? Yeah. 8%. Yes. So 8%. So 8% of people with this was in the programs with, or this was in the, you found it back in the parents.
Yes, so what we did was we had 120 patients with the developmental and philosophy, where we thought it was new in the child, but the routine testing had shown that neither mother nor father had it. And then we looked at each family, we looked at the parents 200 times for their child's specific mutation. And we found that 8% of the 120 patients had one parent who was mosaic, which means there are actually studies of affecting the affected child. And I think there were 10 positive families of the 120, and one of them was KCNQ2, another family with a KCNQ2 family, where the father is normal intellect, works full time, vision stopped at 30, and he has a very severely disabled daughter, and he was 32, and that was in our original paper.
Yes, I remember. Yeah, and I can, or maybe because I didn't really touch upon mosaicism, but because mosaicism, what does it mean is that you carry the mutation not in all your cells. I remember that I said your genes or your DNA has to be copied when the embryo is formed or the baby is formed, and when the mutation occurs later, or this particular person has the mutation in only part of their cell. And it has been shown that sometimes individuals that have a mosaic mutation don't really have epilepsy, so they don't know they're carrying the mutation. But then when they have a child, they can pass it on the variant to their kids and have a kid with, for example, KCNQ2 encephalopathy. And when they have another kid, they can have a second kid. I think it's really important to realize that testing the parents with very deep or very specified techniques to look for mosaicism can help. But it's important to realize that mosaicism sometimes is only present in, for example, the egg cells or the sperm cells of the father or mother. And this is something we cannot detect. So for every child or every parent that has a child with a de novo mutation, it's important that the genetic counselor tells them that it's not 100% excluded to have another child. I think the risk is set in general to be 1%. And then the family should discuss whether there should be a reason for example, prenatal testing or pre-implantation diagnostics. So whatever test you do, you will never be able to fully exclude mosaicism. I think this is the key message. Yes.
So a follow-up regarding mosaicism. So we've got 8% of parents have some sort of mosaicism that sort of passed on the mutation, de novo mutation that they got, or inherited mutation they got. What sort of data and what sort of numbers are there on, for instance, let's say I, as the father, have I've got an 8% chance that if I have that chance, what are the chances that I actually pass that on? So I might perhaps I have an 8% chance of mosaicism, or 4%, I guess, in the two of us. What are the chances that I actually do pass that on versus passing on some of the healthy cells?
Yeah, that's also a very good question, and we actually don't have a correct answer to that. And why is that? Because you cannot predict. For example, you can check in the blood, and imagine that they found that 30% of your blood cells, or only 10% of your blood cells carry the mutation. But we never know how this correlates with the percentage of mutation yield in your sperm cell, right? It might be that only there it's only 1% or even none. So it's difficult to predict what the chance of recurrence is. I think when you know or when a parent knows that he or she is mosaic, they should realize that there is a chance, that's difficult to predict what the chance is. It's lower than 50%, because someone also give a good copy, and probably it's much lower because it's very unlikely that all your sperm or egg cells have the mutation. But it's difficult to put a percentage on it. You should know that there is a risk of recurrence, and then together with the genetic counselor, the medical geneticist, discuss whether or not for a future pregnancy you would like to check this beforehand by prenatal testing or pre-implantation diagnostic.
Sorry, I've got a question as well. It might be convoluted, but is there a risk? Is there a risk that, for example, I have, like monocytism, and so I passed it on. We did prenatal testing for a second, and it came back negative. But would there have been a risk that actually the test would have just another part of the body, and so he would be more sick as well?
That's also a very good question, but there's no chance. And why is that? Because if you would have passed it on, this mosaic variant, it will be in the egg cell that's merged with the sperm cell, and then it's for dividing, and it will be in all the cells. And that's exactly the problem, because you eventually will be right. You would be healthy because you don't have any signs, because the mutation is only in part of yourself, which your kids will have it in all the cells, and you will present problems. Now,
Thank you for your presentation. And we're new to some of this, and so for our non-affected child, then, so our affected child is said to have a de novo. We were told that we didn't have anything, so it sounds like repeat testing is perhaps indicated. So I got that message already. But so for our other child who is not affected, so our non-affected child should also be tested for a mosaic, or how would we? And because he has no developmental issues like ADHD, and so just curious about the non-affected children. How does that impact them to have this mosaic?
no, because there's no risk, and the reason that we're sure is because these disorders are really an advantage to see very early on start where the kids are not have problems, right? Imagine that you would have been, or your husband would have been mosaic and passed mutation on to your kids. Also, your second brother should have the mutation in all itself, and he would have had problems from the start. So if there's no possibility that he carries the mutation, open up mosaic. I mean, if you pass the mutation on, then the mutation is in all itself. And he is healthy, I understand. So there is no risk that he has KCNQ2. This is different for a disorder that only starts later in life, right? So this particular disorder that we're talking about is two-related epilepsy. Children have problems in the first maybe 8 years of life. So if your brother is already older, then there's no need for this. Yes.
I might have one of those huge questions, but how does severity being born the normal cell? But then can these be let's say an exposure to radiation occurs? Like let's say I don't know if you were exposed to all day. There's always a question in our mind. Can these genes mutate? I was born okay, but then I wasn't anymore, and that's something.
It's a complex question. Listen, we do know that certain types of environmental toxic products or radiation, they increase the risk of mutations, right? And if you would, for example, take some brain cells, you will find a lot of mutations that occur during life, also just by becoming older. But they will never be present in all our body. It will be some focal thing, because most of our cells, and especially our nerve cells, they don't keep dividing. They're there from the beginning, and they don't divide, or in general. So if they have a mutation, it stays just in the brain. It doesn't divide into other ultimately. So, for these types of mutations that are present in all body cells before they give problems, it's not like mutation, like cancer or no radiation or whatsoever plays a role. It's different, for example, for cancer, right? Like smoking or whatever, it affects your lung cells, and they accumulate mutations. But this is something very local. But for these kind of disorders, there's no relations. Yeah, that's a good question. I mean, yeah. There's probably, yeah, they say like smoking also affects your fertility and then risk of problems with children. So there's probably risk factors for de novo mutations to occur, but not that you would accumulate it in your cell and be mosaic and then produce it. There's a lot of environmental problems that increase the mutational yield in our cell. By the way, the biggest risk factor for having de novo mutations is should over age of parent, and there is nothing to blame. Just you get older and you accumulate mutations in all cells, and that can be a risk factor. Yeah.
I'm wondering if there's like a possibility of getting it from like she was kind of asking about like radiation, but would like birth control or things like that cause any issues? Our son is de novo and had like a copper birth control when I got pregnant with him, so I was worried about that. Obviously, just wondering if there's things like that that can cause these kind of things.
No, I would like to stress there's no thing that people do that can be blamed for having the organization. It is really nature. And for example, birth control, if birth control make for more, then we should have seen much more genetic disorders in the last 40, 50 years because of birth control. So that's definitely not the case. It's just the fact that from one cell you have to go to, I don't know, how many cells, never know about it. And this is unfortunately that yes.
Question about looking at ethnic diversity and research. My daughter's Chinese, and so we adopted her, and so I'm just kind of curious about what that landscape of research looks like
Landscape of research or landscape
about the week. Do we know anything about incidents or differences among Asian populations?
I don't expect, or we don't expect the influence to be different just because it's a child thing. I mean, Chinese children are made or born at the same way as any other child. There's also there's one child that has to go to, so the incidence of having a limitation is the same all over the world. Obviously, your genetic background and all the variants that you have might maybe influence how kids present. But the disorder, the incidence typically of these disorders that occur due to normal is the same all over the world. It's different for familial things that can sometimes be more present in specific populations because it's transmitted within the population. But the normal patients that occur the same incidence all over the world. So I do not expect it to be different in the Chinese population. I'm sure that there's groups from Asia working on PCP2 too. There might be little contact between Occidental world and researchers from Asia, but there also a lot of research.
One more question. Could you explain the loss of function, gain of function, just a little bit further? Is that alluding to the potassium channel?
Yes.
The channel function or the actual function of the child?
That's the potassium channel. So either the potassium channel doesn't work well enough and you have less fuel to the channel, that's another function. Or the potassium channel is open too much and it's more curing to the channel, and that's the gain of function.
I did.
Yeah, okay, we're gonna have two more questions and then we've got to pause, but remember, we're gonna have a panel with Sarah, with Dr. Beck-Husen, Dr. Schefter, and Dr. Millichap together. So if you don't get, I'm gonna go to you and then further to you, and then we'll pause for now, okay? Because we just don't wanna, we've got a time in here. Thank you so much, Dr. Beck-Husen. Go right ahead.
Along the same lines as the potassium channel, we're trying to understand that, and it sounds like it's circled the pressure to the sodium channel. So some things come in, some things go out. Is it universal and its effect on the in and out?
Yes, so it has, this is really physiology, and it's also not my thing, but the potassium channel, when it opens, the potassium, the ions and the molecules, they always go out. And why is that? Because there's more potassium inside the cell than outside the cell. And when you open the channel, they go out. It's like a diffusion kind of thing. For sodium, it's the exact way, or the other way around, at least in our brains. There's more sodium outside the cell, so if you open the channel, it goes in. So, and this is how our body works, and it's the same for everybody. And it might be different for a channel in your heart or whatever. But for these potassium channels and sodium channels in our brain, that's how it works. Potassium goes out and sodium goes in. Yeah.
Hi. My husband has epilepsy, but my children do not. But we have a KCNQ2. One is a child. Does it ever skip generations?
Talking about KCNQ2, well, I suppose your husband doesn't have KCNQ2 or doesn't have the neonatal epilepsy, and let me put it another way. Epilepsy is very common. So epilepsy in general, it's present in 1 in 100 to 1 in 150 people. So looking here in the room and regarding the kids, there will be apparently other people with epilepsy there. That should not always be related to KCNQ2 related epilepsy. If, and I don't know the story of your grandchild, this is the generation, but I expect it's a genome mutation, so then it has nothing to do with the epilepsy in the grandfather or healthy grandfather. Epilepsy is very common, so you will even in family that's a problem or epilepsy due to genome mutations, you might have had a family with epilepsy.
All right, and I think we're going to pause right there. Dr. Beck-Husen, thank you so much for meeting with me. As we're switching over, I did want to mention one thing just before you turn off your mic. We're learning more all the time. And I just wanted to say, if anybody has other questions, remember that there's going to be a genetic counselor here tomorrow. And you might just have another couple questions that you want to ask. So feel free to do so. And the other thing is, you mentioned this, but can you just say one more thing about luck? Because, I heard questions and I'm thinking, it's so easy as a parent to blame yourself and say, I did this, I didn't do that. And this, can you just talk about this is random?
I think this has also been the great, I don't know if advantage is a big word of genetics. These de novo mutations, as I said, they occur. It's nature. It's bad luck. And I've seen this, and I think many of my clinician colleagues here in the room might echo that. There's so many families that I've seen that have blamed themselves, and particularly moms, but probably also dads, that said, I drank this glass of alcohol during pregnancy, or it was this or that, or this pill that I took because I had the flu or whatever. maybe this caused the problems in my child. That was not the problem. The problem was just bad luck, and that's what genetics has. Besides from the fact that we know what it is, and hopefully we find a cure, is that I hope that it can take away some of the blame that families are, and particularly mothers, but I speak as a mother maybe, that they take on them. It's not because something what you did is just, again, it's a miracle that it doesn't happen more often. You go from one cell to, I don't know, millions of cells, and sometimes there's a mutation that occurs, unfortunately.
Dr. Wekhuizen, thank you so much. I appreciate that.